First of all, welcome to everyone to this month's BBI seminar. We're really fortunate this month to have Elizabeth Strychalski, who will introduce in a little bit later. For those of you that are not familiar with the way we run these seminars, so we'll have a talk, and if you have any questions during that talk, we make use of another website called Slido. So what I'll do is I'll post into the chat the Slido meeting number. So if you Google Slido and then search for that website, you'll be able to then go on that, type in this number, and be able to post questions, and you can then upvote questions that you would like asked or ask your own questions. And what will happen is myself and Lucia Marucci, who's helping me co-host today, will actually field the questions to Elizabeth so that we can then make sure that all of those are answered and processed. So I guess without any further ado, I'm going to hand over to Lucia now, who can introduce our speaker for this month.
Thanks, Tom, and thanks, Elizabeth, for being with us today. So Elizabeth founded and leads the Cellular Engineering Group at the National Institute of Standards and Technology, that is the National Metrology Institute of the US. Her group provides a foundation of high-quality measurement tools and methods to support the design and control of biological function for applications of engineering biology. Her group participates in a number of community groups, such as BDCell, SBOL, the Global Biofoundry Alliance, and the Engineering Biology Research Consortium in the US. She also served as a program manager in the Biological Technologies offices at the Defense Advanced Research Projects Agency. There, she managed a portfolio to advance experimental and theoretical research at the interface of synthetic biology, control engineering, nanobiotechnology, and fluidic devices. She received a number of awards, such as the University of Rochester's Harry Fulbright Prize and the Janet Howell Clark Award, a National Research Council Postdoctoral Research Associateship, and the Maryland Academy of Sciences Young Scientist Award. So, Elizabeth, we are really keen to hear from you now.
Thank you so much. Let me share my screen here. And okay, so thank you. It's a real pleasure to be with you here today. I do need to start with a disclaimer that anything we discuss today does not imply a recommendation or endorsement by NIST. And so you can understand my background and biases. I've spent time learning and thinking about physics, atomic and molecular optics, high energy physics, nanotechnology, analytical chemistry, and now finally, synthetic and engineering biology. But more than any one of these areas, I like to think about metrology, which is the science of assessing value. So how can we use biology itself to construct measurement tools for living systems? But before we answer that, I'd like to first briefly consider the impacts and obligations brought about by biotechnology. So we all started as a single cell, like this image on the left, and this is of course utterly ordinary. And the image on the right is an electron micrograph of genomically minimal cell, JCVI SYN 3.0. This is extraordinary. This is a human achievement at the forefront of our ability to engineer and understand cellular life. And for me, these summarize two truths. One, that we're biological, and two, that we're biotechnological. Now, we already know from experience that we're capable of getting ourselves into existential trouble through our technology, and there's really no reason to think that biology should be any different. The issues of what we could do, what we should do, and what we will do are at the hearts of developments in biotechnology today, and the future that we choose to make for ourselves. And I think it's important to start by claiming ownership of our tremendous responsibility in this. So biotechnology is awesome, but it's also very ordinary. You know, consider something we take entirely for granted, commercial passenger flight. This might be routine, but we don't have to become routinized to the fact that we sit calmly in chairs, hurtling through the literal stratosphere. And the impacts of this are so much more than just putting people in chairs in the sky. Personally, I find biotechnology to be much like this. We all know here that transforming genes between organisms, like air travel, is entirely routine, but we don't have to become routinized to the fact that we can, with intent, read and write the very DNA expressed inside living cells. And the impact is already so much more than fluorescent fish in your home aquarium. Take CAR T cancer therapies, for example. And by the way, these transgenic fish have been found in the wild in Brazil, which highlights issues of biocontainment. So the benefits and perils of biotechnology all play out together to impact our daily lives. And we can consider the schematic that shows what are generally agreed to be the various technological revolutions of mankind. I should say humankind. Steam engines, mass production, the automation, computing, and information revolution, and then where we live today, the artificial intelligence and Internet of Things revolution. And then there's this last one where we find ourselves now in our discussion of living measurement systems in minimal cells. Some argue that we're seeing the development of the foundational technologies that will drive the next technological revolution, the biotech revolution. And synthetic and engineering biology are, of course, major drivers of this. It's interesting that one measure of a technology's success is our ability to take it for granted in our lives as technological beings. So where is all this biotechnological capability going to take us in the future? Now, we all know that most engineering biology still happens in laboratory settings. The future, we'll see these move increasingly to use outside the laboratory. And I think it has to be that way because I'm hoping that we can field these technologies in time to solve pressing challenges for our environment and for human health, for nothing less than to ensure our long-term survival. And if we can allow ourselves some freedom to dream, we can envision influencing entire planetary systems to deliberately better support human life, which is the opposite of what we're doing today. Although this is meant to be Mars in this image, we can imagine proving out terraforming capabilities on smaller targets like comets or asteroids. And hopefully we'll get a chance to do this before we have a dire need for those technologies on Earth. My point with all this is that I don't think we should limit our imagination when considering the potential of biotechnology to transform ourselves and our environment. And we can only build the future that we can envision together. This takes us right back to issues of what we could do, what we should do, and what we will do. And I believe that we all have the responsibility to ourselves and to each other to help shape this, because we all want a future that's worth living, and we don't want to leave anyone behind. Okay, stepping off my soapbox. These are the salient points of our discussion today that I hope you'll take away with you. One, that there are institutions and people out there to help you with your measurements. Two, that we can use parts from living systems to build measurement tools. Three, that we now have the ability not only to bring better measurement tools to cells, but we can alter cells to make them more amenable to measurement and engineering. And four, between genotype and phenotype, there's a lot of opportunity to use simple physical models to better understand the physiology of minimal cells and to attribute function to genes of unknown function to reach that tantalizing goal of one day knowing the function of every gene in a cell. Okay, so starting here, my group lives at NIST, which is the National Metrological Institute of the United States. And it's part of the Department of Commerce, because if you can't agree on weights and measures, you can't trade. And a core part of our mission is to focus on metrology, which is again that study of assessing value. I happen to be located in the material measurement laboratory where our engineering biology program is centered. But research supporting measurements, equipment, data management relative to biological systems occurs in nearly all of these laboratories. Now, people often associate NIST only with standards, but I invite you to consider the incredible amount of technical work and stakeholder engagement that has to occur well before we come to the creation and use of the standard. NIST is non-regulatory, so everyone using a standard has to agree that that's the standard we're all going to use. Now, as a technology area matures, we tend to build up this pyramid. And engineering and synthetic biology, as rapidly developing fields, tend to still inhabit mostly the lower parts of this pyramid, which is where my group does a lot of their work. It's rarely too soon to engage with your local friendly metrologists about your personal measurement needs and what you see as the broader needs for your field of expertise. And it's important to do this because meeting these needs as early as possible will speed filling out this pyramid, which will in turn support the success of your research, your startup company, and the bioeconomy more broadly. Now, NIST is developing measurement products that are enabling the predictive design, engineering, manufacturing, and translation of emerging biotechnologies. And I like to think of the road to realizing the future bioeconomy as paved with measurement innovation and measurement assurance for both biology and engineering biology, because they're generally advancing together from discovery and innovation to knowledge and capability, and finally prediction and control. And I know that all of us here can appreciate just how vital control is to achieving this vision for engineering biology broadly, that is the routine ability to design and build safe, well-characterized, and predictive function into biological systems for a desired purpose. Now, to remind you of the impact of control, we can consider the difference between building walls by piling up rocks you find while clearing land for your cows to graze and delivering water to Roman cities, or giving expression to human artistry by creating these beautiful arches in the Hagia Sophia. The difference is from an incidental qualitative outcome to a deliberate quantitative outcome. So how do we arrive at better control to make custom biological parts built for purpose? Because you can't tell me that you can guarantee that you're going to find something like the perfect keystone when you're clearing land for your cows to graze. So once you have the right part, then how do we control what the parts do individually and in concert with each other and in context? So to support this objective, we founded this group at NIST with the mission of providing a foundation of good measurements to support control and rational design of biological function, driving towards something that we're calling precision engineering biology. So basically, we want to control the life force. Okay, so that's who we are, but what are we actually doing in the laboratory? Well, we develop living measurement systems to enable control. These use biological parts to measure biology and enable the implementation of control strategies in living systems. Now, I would argue this is the right technology for the job. It's compatible and scales favorably with the biological systems we're trying to measure and engineer. And we know it's possible because biology is measuring and controlling itself all the time at every level. I want to briefly introduce 3 areas my group is working in to advance living measurement systems. And this will lead us to our discussion of minimal cells. So the first area is sensors. If you can't sense, then you don't know what your system is doing and you can't correct that function or output. In other words, you can't implement control. The second area is computation. Processing information from multiple inputs, maybe coming from the environment or internal to a cell, can greatly increase your options for control, for actuation, for reading out measurement results, and so on. And 3, chassis. We use typical various lab strains of E. coli and yeast. We also use cell-free systems and genomically minimal cells to come with a problem of what is life, how can we control it, and what are the boundaries of what it can do for us? So stated differently, those questions are, what is a biological system? What is its identity? What can it do? What is its function? And what could it do? What are the possibilities or opportunities for engineering? So to support our sensor work, this is NIST's first biofoundry, the Living Measurement Systems Foundry. This is part of our larger push to modernize and automate our biological labs at NIST. This is a flexible system designed currently around testing genetic libraries in bacteria and yeast. And ultimately, we want to drive our automation in a loop where our high throughput measurement results feed into machine learning methods we're developing that then make predictions and design the next iteration of our experiments. But we found so far that we've gotten so much high quality data from just one iteration that we haven't yet had to close that loop and do more. David Ross in my group leads this work, and he currently applies automated workflows to deliver allosteric protein sensors for use in bacteria and GPCR sensors in yeast. He has plans for expanding to other types of protein sensors in organisms. So with this work, we started with a directed evolution workflow, adding a step, because we're NIST, where we measure everything. So to remind you, directed evolution involves creating large libraries of many variants of a DNA sequence encoding for some biological molecule or a function of interest. Now, instead of the usual workflow where you select only a few winners and throw away the rest, we decided we were going to measure the performance of every one of those variants in the library. So we constructed a large library of randomly modified DNA sequences encoding for slightly different versions of our protein sensor of interest. And to prove out our workflow, we chose LAC-I simply because this is a well-known model system in E. coli. So we put these into our bacteria and we measured the response of each sensor to many concentrations of an inducer molecule. So this project had 2 main outcomes. One, by measuring everything, we can discover novel function. That is, we can find what we weren't looking for or didn't know to look for, or maybe never saw in nature. For example, this project rediscovered what we're calling band stop sensors, which as far as we could tell had been forgotten in the literature. They were certainly unknown to us when we started this project. It wasn't until afterwards when we went back and really went digging that we realized these had been known before. The second outcome of this project is to enable precision engineering, either by simply mining the data and choosing DNA sequences in silico that match your desired performance requirements for your sensor. Or you can forward engineer by learning sequence function design rules using machine learning and then predicting the DNA sequence that gives you the sensor you want. I also want to note that we're publishing our detailed protocols on Protocols.io, and we're sharing our automation and machine learning methods on GitHub. And we're also a member of the Global Biofoundry Alliance, and we're very happy to share our experience and advice on automating synthetic biology protocols. One recent machine learning success from our sensor work is called Lantern, which will come out any day now in PNAS. It's a hierarchical Bayesian model that distills genotype-phenotype landscape measurements into a low-dimensional feature space that represents fundamental biological mechanisms of the system. And this is a really exciting development because it eliminates the trade-off between interpretability and predictive accuracy that you so often see in machine learning. I realize I'm going really fast. Please feel free to ask questions about any of this. Moving from sensors to computation, we demonstrated co-transcriptional RNA strand displacement circuits. These take the power and programmability of DNA computing and instantiate it as genetically encoded RNA circuits based on toehold strand displacement. Our circuits can be transcribed in situ and continuously in a way that wasn't possible before and is compatible with use in vivo. You can imagine then a cell monitoring its internal state in real time for all of the reasons you might want to do this for cells making products or for cells themselves as products. The idea for us is to take the entire measurement pipeline and move it out of our laboratories and out of our in silico computers and instead use biomolecules to build that measurement pipeline inside a living cell. So we were able to demonstrate single layer Boolean logic and amplification circuits in vitro. And we've also demonstrated multi-layer cascades. There's also a simple model that allows straightforward prediction and design of circuit performance, and that code is available on Git. We're working now on testing design rules for these circuits to enable more complex computations and hopefully do some computations that move beyond Boolean logic, which we all know is not how biology likes to do its business. Oh, I'm sorry. Okay. So regarding chassis, one of the types of biological systems we work with are cell-free systems. These are reactions in which you essentially take the innards of a population of cells, often E. coli, remove the DNA, and add your chosen DNA template along with any other additional reagents you might need. Now, there are three common reasons why you might be using cell-free systems. You might want to use it as a platform in itself so that you can harness the functionality of biology without that system needing to be fully alive. Or you might want to use a cell-free system as an intermediate tool for testing, say, genetic parts and speeding your development workflow. You might also be trying to construct something like a synthetic cell. So we're working to provide measurement support for all three of these use cases. Reproducibility has been a challenge for cell-free systems in research settings for a number of reasons. We've focused recently on testing manual and automated protocols for preparing the DNA template and quantifying the resulting amount and variability of protein expression. Now, interestingly, we found that automation is not always a better option than a manual process to reduce variability in protein production. So automation is not always better. And now we're also developing in-house automated methods for lysate preparation and encapsulation to speed part testing and validation. We want to enable testing a comparison of the function of engineered biological parts anywhere along the spectrum from chemistry to cellular life. For example, we hope to develop a reporter scheme that works across all of these systems to help us take our RNA circuits in vivo as a measurement tool. And we also want to be able to share this capability with other laboratories who've expressed a need for this, this kind of a measurement tool. Okay, so those are some projects happening in the Cellular Engineering Group. I'm happy to go into more detail on any of them. But now I'd like to move on to minimal cellular life, which I hope to engineer into a platform for developing and testing living measurement systems. We've been involved in creating and characterizing the chassis of genomically minimal cells. So specifically, that's JCVI Syn3.0, Syn3A, and related strains. You know, the minimal cell has been a dream of the scientific community for nearly 100 years. A group of scientists formed the Phage Society in the 1930s and thought deeply on the concept of a minimal cell. And then later in the 1980s, mycoplasmas were suggested as an ideal, naturally evolved cell type to use as a starting point for creating the minimal cell. So researchers have been asking, can we throw away functions? Can we throw away biomolecular parts until only the essentials remain? What would be left? What does a minimal cell still have to accomplish to stay alive? So here's a list of essential cellular processes according to John Glass at the JCVI. That's the J. Craig Venter Institute. And he led the development of the minimal cell we'll be talking about today. And what I love about this list is that you'll notice that adaptation or evolution is not on here. Another set of questions we can ask is, what might minimal cells tell us about the origins and evolution of life, such as our last unicellular common ancestor? And conversely, how can we use what we learn about phylogeny to inform our ability to better engineer synthetic and minimal cells? Now, personally, I find it helpful to situate the minimal cell today within a broader landscape of cellular engineering. And this landscape, you can consider it as having 4 types of cells. You have naturally evolved life. These are the organisms you find out in nature. You then have cells you've edited intentionally. You've imposed some rational design on their genome. You can also use biological processes to screen and grow, for example, using one of these DNA libraries, many different variants of a function, and then simply pick the one you want to propagate further. And then there's synthetic cells or synthesized cells. Sometimes it's called lineage agnostic life. And this is where, in principle, you have full mastery of cellular life, which we do not have yet. So focusing in on that, oh, and I should say that those types of cells are not mutually exclusive. But focusing in on the last category, those synthetic cells, there are generally considered to be 2 main approaches for creating synthetic cells. Some researchers use a bottom-up approach, which has not yet, to my knowledge, been fully realized experimentally. Now, JCVI's minimal cells are instead examples of the top-down approach, where the idea is to remove parts of an already functioning cell until it just barely meets some definition of life that you've set. Now, moving forward, there's tremendous opportunity in also combining these approaches. So the minimal cell was only a reductionist fantasy until it was no longer a fantasy. So how did we get there? Like, what capabilities were needed to arrive at JCVI's minimal cells through this top-down approach? Well, they developed several technologies that together enabled building cells with synthetic and minimized genomes. Now, first, genome synthesis was published by Dan Gibson and colleagues in 2010. So genomes were synthesized from oligonucleotides, and the last assembly steps were done in yeast. These genomes, importantly, did not have to pass through any viable intermediates. So this allows for large genetic changes. Second, genome transplantation was published by Carol Lartigue and colleagues in 2009. So the donor genome is isolated from a yeast nucleus and then transplanted into a phylogenetically related bacterium. Recombination is blocked between the donor and recipient so that we can select for only the donor genome. So then, because only that donor genome is there, all the new proteins in that cell are then encoded from the donor genome. So within something like 10 generations, all proteins in that recipient cell are now encoded by the donor genome. And in this way, that donor genome reprograms effectively the recipient cell. Now, third, they use an engineering workflow based on design, build, test or design, build, test, learn cycles to engineer with synthetic genomes. So in 2016, Clyde Hutchinson and colleagues, including NIST, used this approach to develop a cell with a minimal genome. Now, NIST was involved specifically in providing measurements on genome transplantation and cell phenotype. So we can't take credit for any of the rest of it. Okay, so these capabilities were applied to Mycoplasma mycoides. So again, mycoplasmas were suggested in a paper in 1984 as an appropriate organism for genome minimization because they came pre-minimized by nature through a loss of genetic complexity during adaptation to life as an obligate parasites. So these are opportunistic pathogens of goats, which makes for a hilarious situation when you have lab tours because you have to ask people if they have pet goats at home. They import complex metabolites from the host, so they don't need any machinery to make them for themselves. They have naturally small genomes. They do require a very complex growth medium. So it's sort of a trade-off between, like, yes, you have a minimal cell, but then it needs sort of a maximal growth medium. So you have to make that trade-off. Now, importantly, they do not have a cell wall. So that'll come up again later. Now, for a sense of scale, here's a Mycoplasma mycoides shown roughly to scale with E. coli and bacteriophage. So they're very small, and this also poses experimental challenges. Okay, so first, a version of the Mycoplasma mycoides genome was synthesized and transplanted into the related Mycoplasma species capricolum, and that gave us JCVI SYN 1.0. This was, back then they were calling it the synthetic cell. I don't think that by today's definition of synthetic cell, we would be using that vocabulary. The DNA sequence of 1.0 largely mirrors the sequence of wild-type Mycoplasma mycoides. Now, a subset of the genome of JCVI SYN 1.0 gives us SYN 3.0. Now, by the way, there was a 2.0 during the minimization process. In fact, there were many, many strains. And given the state of our knowledge, much of our workflow was, and it had to be, empirical. So there was a lot of just trial and error, testing different combinations of genes included in the genome, genes taken out of the genome. But why was the minimal cell developed using 8 genome segments shown in the schematic? So this is something that I asked Dan Gibson, and I said, why 8? Why not 7 or 9? And this is one of those things in science where there was no good reason. It was just, that was a convenient segment size to work with. But it also let them deal with something called synthetic lethality, which is a major challenge for genome minimization, and it reflects how gene essentiality depends on genomic context. So synthetic lethality can happen when multiple genes redundantly serve the same function. So consider an airplane with 2 engines. Removing either one of its engines still allows that plane to fly. So you might consider the engines as non-essential. However, if you simultaneously remove both engines, your plane can't fly. That's synthetic lethality. So they were able to test each one of these segments separately and in different combinations to try to understand the synthetic and manage synthetic lethality. Okay, so we can compare the growth of SYN 1.0 and SYN 3.0, and we see that 3.0 had a slower but still reasonable doubling time, and it produced colonies that are morphologically similar to those of JCVI SYN 1.0. So 3.0 was optimized for minimal genome size, with the constraint of fast growth at the population scale and confirmed by population scale measurements. This then inadvertently left whatever was happening at the single cell scale essentially completely unconstrained. And this happened. So this scanning electron micrograph confirms that single cell morphology was not constrained during optimization. And you can see that SYN 3.0 at the single cell scale was much more heterogeneous than SYN 1.0. Now, fundamentally, this raises interesting questions around the connection between genotype and cell division and growth. For example, control of cell morphology is evidently not a requirement for genomically minimal life. Who knew? I mean, what other assumptions or biases might be wrong? Now, practically, this poses problems associated with trying to study and engineer cells that don't have reproducible characteristics at the single cell scale. So JCVI and their collaborators asked NIST for a way to measure the dynamics of single cell growth and investigate what genes are required for a normal morphology. So we developed microfluidic chemostats for this purpose. We deliberately kept the fluidic devices very simple because measuring biology is hard enough. These devices consisted of a single main microchannel with many smaller growth chambers along the length of that main channel for cell culture and imaging, and there were no moving parts. These were molded into polydimethyl siloxane, PDMS, bonded to cover glass, and imaged on a wide field epifluorescence microscope overnight. Just like the mother machine devices developed for E. coli, which you can see here on the left, small molecules such as food and waste simply diffuse between our main channel and the growth chambers over a period of seconds. Now, it's important to remember that our devices enable these cells to grow in a shear-free environment because these cells have no cell wall, and we wanted to be sure that any morphologies we see result from processes intrinsic to the cell and not because of anything external imposed by that extracellular environment. So to give you a sense of scale, here's a typical mycoplasma colony on an agar plate, shown actual size with one of our microfluidic chambers. So the colony is that big dark orange thing. Our microfluidic chemostats then extend our ability to engineer the minimal cell, not only at the colony scale, but also the single cell scale. And for those of you accustomed to looking at huge cells like mammalian cells or even E. coli under light microscopes, I really want to emphasize the scale bars here. The minimal cell is quite small, comparable to the wavelengths of light we use for imaging. And our chambers are, I believe, 3 microns deep. And so the cells are also diffusing in and out of the depth of focus. So it's not that we're bad at microscopy, it's that things are really small. So how did our cells grow in the microfluidic chemostats? As a control, these time-lapse images show growth of JCVI Syn 1.0 cells. They appear to have a small, uniform shape. They diffuse freely within the chambers. And you can see that here in this phase contrast image, along with an image showing constitutively expressed fluorescent M cherry in the cytoplasm. That's a fluorescent protein at each time point. So you can see this is happening during overnight growth. I always get the question, why do the cells seem to collect at one side of the chamber? So on the right, there's the neck of the chamber leading to that main channel. And on the left, the cells seem to collect. And that's because there's a very small flow of water into the PDMS. So your buffers, your liquids will evaporate through your PDMS over time. Now, in contrast, JCVI Syn 3.0 shows a lot of morphological variation, including cellular structures that are several microns in diameter. You see filamentous cells, you see other shapes, you see things that may or may not actually be cells. Maybe there's some kinds of vesicles. And so I'm going to play this and then kind of flip back to the other. So this is 1.0. And then 3.0. And keep in mind, these are just representative chambers. We have thousands of these chambers that we're watching. Now, in addition to variation in cell size and shape, Syn 3.0 also varies in biomolecular composition. So for example, some have more or less DNA, some lack metabolic activity to express M cherry. At the end of the growth experiments, we can flow fluorescent labels in to visualize, for example, the DNA and the membranes. We can add fluorescent media additives to test membrane permeability. There's a lot of potential to use our chemostats to learn more about the minimal cell phenotype. But for now, I want to focus in on which genes are required for normal cell division. So we hypothesized that Syn 3.0 lacks the genes it needs to control division. They were simply taken out as non-essential. Now, there is an alternative that adding or overexpressing genes could also perturb normal cell division. I just want to admit that. I'm going to put that on the side. We first tested and minimized versions of those 1/8 genome segments in the context of an otherwise unminimized JCVI Syn 1.0 genome. Now, only the minimized segment 6, you can see that in the lower left here, caused abnormal cell division consistent with what we saw with 3.0. So this restricted our search from 428 genes removed from 1.0 to make 3.0 down to 76 genes missing in segment 6. So this was hugely advantageous to have identified that. Now here's a striking video showing the overnight propagation of one of those cells with a minimized segment 6 in the genomic context of Syn 1.0. I want to let this loop a couple times because this is maybe my favorite video of the whole experiment. It's just amazing. So we wanted to get to the bottom of this and really understand what gives rise to these morphologies. But so by creating many versions of segment 6, each with different combinations of segment 6, each with different combinations of those 76 genes removed to make Syn 3.0, we arrived empirically at JCVI Syn 3A. Now I bring this strain up explicitly because this is the most widely used strain among the dozens of labs doing research with the minimal cell. I think it's up to 54 labs or something. This strain retains 19 genes, not in the minimal version of segment 6, and it gives us nearly normal cell division. So we were able to rescue that JCVI Syn 1 phenotype. And here's another video showing that reduced segment 6 in the 1.0 genomic context, and then SYN3A. So not totally normal, nearly normal. So these 19 genes added back to SYN 3.0 were organized into 8 clusters. And in each cluster, genes were adjacent and could represent transcriptional units. So we studied those intact clusters before we studied them individually. Now, we first hypothesized that cell division depended on a gene cluster that's part of the highly conserved division and cell wall cluster involved in cell division in many bacteria. This DCW cluster includes the tubulin homologue, FTSC, which is known to form filaments that localize to the division site in many bacteria. So we thought that just adding this cluster alone would restore normal cell division. But to our surprise, it did not. The cells still lacked size and shape control. What's more, we're looking at the annotations for the other genes, and this didn't offer us any hints for which genes might be required for normal cell division. So we kind of had to take a step back and say, is there a more systematic approach that we can use to build strains and test them with different combinations of these 19 genes? And Li Sun, who was then at JCVI, did this. She used several genome engineering techniques to test different combinations of genes, first in clusters and then individually, until through this comprehensive search, we were able to identify 7 genes. Now, what I love about these 7 genes is, so there are 2 with a known cell division roles, FTSC and CEPF, but there's also a hydrolase of unknown substrate, and there are 4 genes that encode membrane-associated proteins, but with unknown function. And we know that these are all required together to restore a normal phenotype, similar to JCVI SYN 1.0. Now, it makes sense that cell division requires changes to the membrane, but the molecular mechanisms remain unclear. Our study really wasn't designed to pin down the mechanisms. We wanted to identify genes. So we were able to connect this genotype in the minimal cell to the phenotype of normal cell division, but none of this reverse genetics approach informs us about the molecular mechanisms of gene products that then result in that phenotype. So how can we generate hypotheses for how these genes might contribute to normal cell division in this organism? How can we get a better understanding of what's happening? Now, there are many possible formalisms and frameworks that could help us understand relationships between genotype and phenotypes in minimal cells. And we can kind of set the genes aside. We can take a step back and consider the cell as a physical object composed of gene products. For example, we can ask what mechanical forces acting on the membrane could drive constriction and scission during cell division. Now, we know that there are models for vesicles that can provide a simple starting point for membrane mechanics. These shapes were adapted from a model for vesicles that predicts, under certain conditions, vesicles can constrict and divide in the absence of any specialized machinery, such as FTSC. If this can be true for vesicles, then why not for a cell? Why not for our minimal cell, which lacks any confirmed division mechanism? In other words, how much of the cell division is governed by the physics of the cell as a collection of biomolecules, such as a material, versus being driven as an active process regulated by the cell in a way that consumes energy? So there is one very simple model called the spontaneous curvature model for vesicles that we can consider cell division of SYN3A within the context of. And there are 2 key physical parameters in this model. There's the volume to surface area, and there's the spontaneous curvature of the membrane. So we're looking for insight into which genes these parameters might depend on in this genomically minimal context. And hopefully this can give us information on which gene products are contributing to cell division. Maybe it can help us to start assigning functions to some of these remaining genes of unknown function. So this is just schematically where our understanding currently is in terms of which genes might be playing a role in these 2 parameters. You can see a lot of question marks because there are still a lot of questions. So what's next? What else are people learning about the minimal cell? This is a very exciting time in the field. We have whole cell models of the minimal cell. Others are looking into its evolution. Still others are putting functions back into the minimal cell that it never had to begin with, such as the ability to move. There are a lot of papers coming out now or about to come out. We're part of Build a Cell. If you're curious to learn more about synthetic cells and the research that's happening to make them a reality, this is a very welcoming, supportive group. There are also exciting opportunities, I think, for minimal cells as test beds for research and development, for biomanufacturing, for cell-based products. And, you know, at the end of the day, no matter where we go with this particular minimal cell, building synthetic cells in general will inform and ultimately require total mastery over cellular life, which seems like a really formidable challenge. So why bother? Now, some of us are driven by curiosity to take up challenges because we can, sort of like climbing Mount Everest. We're sort of driven to discover what constitutes life and its inner workings. Other people see the need for biotechnologies to solve pressing societal problems that require the ability to control and engineer cellular function. And some others might want to use biotechnology as a lens to more fully experience who we are and how we can have a meaningful life. You know, we even have a popular mythology in which the life force plays a central role, and there are cell-based transducers of this force. And once we have full mastery over the life force, then what else might we aspire to? You know, we can start asking questions like, how can we take what we learn about life from the minimal cell and apply it to create living or lifelike attributes in non-biological systems? How might we go from machines like Atlas to machines with key attributes of life? Like the character's data or the Tin Man, you know, what would we gain? What would we lose? What are our responsibilities to our creations? What are their obligations to us? And who gets to access all this technology? Those are questions for another day. For now, I'd like to thank all of my collaborators who have been fantastic to work with. And I'd like to thank you for your attention. I do want to mention that my group does have open positions and we're always open to new collaborations. Thank you so much for your attention.
Thank you so much, Elizabeth. That was a real tour de force of all the different things that you're working on, which is great. That was absolutely brilliant. We've actually got quite a few questions in the Slido, so I'm going to get started straight away, and me and Lucia will just bounce back between each other. So at the top of the list at the moment, we've got a question around, where do you think the sweet spot is for synthetic cells between a minimal genome and robustness and survivability? So kind of how much more than a minimal genome do you need before it becomes really exciting from an engineering perspective, I guess?
That is a fantastic question. And that is exactly, I think, where we need to focus some attention. And as soon as you start talking about engineering, you're talking about doing something for some purpose. And so you'd want your biological system to be fit for your purpose. So ideally, an engineer would be able to walk up to a spectrum from non-living to living, you know, from chemistry to biology, and say, well, my system needs to have this biological functionality, but doesn't need all the rest of it. You know, you want to be able to have a suite of systems to choose from, so that you're engineering with only as much complexity as you need for your purpose, and not any more that's going to muck up what you're trying to do.
Yeah, that's great. Lucia, do you want to?
Thanks, Elizabeth. There is a question that is, I think, about your time lapses. So do you think there is any density-dependent behavior in your GCVI scene cells? And actually, I wanted to ask if you also think there is any effect of the geometry of your PDMS chambers.
Great questions. So it is possible that there is an effect as the cells grow into the small population of the chambers and start to clump. I will say that the cells look to us, to the resolution of our measurements, as they do in growth outside of the chambers. So we did do comparisons side by side. We wanted to make sure that what we were seeing in the chambers was at least comparable to growth on the benchtop. With regard to effects of the geometry, I think that is such an exciting question. And it sort of pained me as a nanofluidicist by training to make microscale devices for this measurement. Knowing that it really wouldn't be difficult for me to implement a device that would match the dimensions of the cell to really enable, to stop it from moving around so much. These are non-adherent cells, to do better microscopy. But for the purposes of this, this set of measurements, we didn't want to constrain them mechanically too much, because we were worried, without a cell wall, that would influence not just the shape, but who knows, maybe even the gene expression. I don't know. But lots of opportunities there.
So next on the list is, I think this is from earlier in your talk where you're talking about some of the band stop sensors. If you could elaborate on what a band stop sensor is and kind of its basic functionality and how it works, I guess.
Also an excellent question. So we were expecting, let's see if I can draw this. We expect sensors that behave like, what, is it going to work?
Oh no, it's
no. Where if you have the inducer concentration, if there's like sort of amount of expression on the y-axis, inducer concentration, something that looks sort of like sigmoidal, where there's an off state and a ramp up to an on state. You can also have inverted sensors that kind of start on and then turn off. We were surprised to find some that start on as you go up in inducer concentration, turn off, and then as you go up further in inducer concentration, turn back on. And the PI, David Ross, on this work, and I both come from a physics background. And so we're used to thinking about notch filters and, band pass, band gap, whatever, so that's sort of the language that we had picked up to describe this, this behavior.
Is that something that's used in nature, or is that one of those, if you look at natural sensors, do you see those sorts of behaviors, or is it one of those things that you just discovered, is the capabilities there, but we just haven't seen or even looked into that, I guess?
Personally, I'm not aware of any in nature. I would love to be proven wrong. As a physicist, we're used to learning that if something can happen, it does happen. But biology is not physics. So is there a reason why biology doesn't seem to use it?
Yeah, it's crazy.
Thanks. So the next question is, do you think that standards in synthetic biology will become a commonplace as in other engineering disciplines, such as computing? Or do you hope that maybe?
I think they will. I think we don't know what they will look like yet. And I think computing is a great analogy, because there are a lot of standards that people agree on that have enabled tremendous progress and industry. But you can also go to a semiconductor electronics user fabrication and make whatever you want. Just because a standard exists doesn't mean that you have to use it, right? Remember that at least in the United States, we're non-regulatory. We can't tell you what to do. Now, that may mean that you won't get approval by the FDA. You may not be able to prove safety and efficacy, or you may have a product that's not compatible with other technologies that the consumer might want or find useful. So, there are consequences to decisions you make around standards, but remember that standards come from a place of everyone, all the stakeholders sitting together at a table, sometimes quite literally sitting together at the table and saying, let's agree on how we can do things in a pre-competitive way that will enable all of us.
I think that's a really good comment for sure, because I think people get kind of nervous around standards. It's like they're signing up to something and they can never go outside that box that they've now signed up to, which is really not true at all, but super important to understand. I don't know if it's not really a related question, but it comes back to the kind of the measurement technologies you're talking about, in terms of how do you know that we're measuring the right things with biology and not just the things that are easy to measure? Which I think is a really good question, because I think as a physicist, there are certain things in the world that we know about, but are there things in biology that we don't know about that we should be measuring that perhaps you're thinking about?
Wow, there's a lot to unpack there. I think we're very much aware of this challenge. We walk around with hammers and find a lot of nails, but that doesn't mean we should be looking for nails. or that we should be walking around with hammers. It just happens to be the tool we have. I think that we don't know yet what the most meaningful enabling measurements are for biological systems. I think we need to sort of on a case-by-case basis decide for our purpose, what measurement is fit for purpose. I can say that we do think carefully about, for example, for our RNA circuits is a good example. If we want to query how our RNA circuits are performing, do we really want to wait for a fluorescent protein to start folding to give us our signal? Or do we want to find a measurement that's sort of closer in time and in, I guess, central dogma space, to that behavior that we want to know about. So issues of what is your measurand versus what is your measurement is key here. I agree.
Actually, I had a related question, Elizabeth. So in the first part of your talk, are the majority of your measurements taken at steady state, or do you measure also the transient responses of cells?
Good question. So there are a couple of answers to that. Most of the measurements are done for the sensor work in exponential growth, I believe. But I want to comment also that our automation setup is a little bit different, because when you walk up to our automation suite, we don't have a big incubation module off on the side where we slot in a bunch of plates. We instead incubate in our plate readers so that we can take measurements during the whole growth process of our cells. So we're high throughput in terms of measuring those DNA libraries. We're not high throughput in terms of the number of plates per hour, that we're looking at. So it's a little bit different. And so we have, I think, 6 plate readers currently. 4 of them measure only absorbance, and then 2 also have the capability for looking at fluorescence.
Thanks. Another question in Slido is about your cell-free systems. So they're asking, are you doing anything around fully reconstituted cell-free systems where all the components are known?
Right. So these would be like the pure systems, the pure express systems. Yeah. And right now, we're not married to any particular system. We're trying to understand stakeholder needs. And right now, a lot of people are using these reconstituted systems. And so I would say so far, we've mostly used those systems as a first stepping stone from going towards lysate-based cell-free systems. And then we want to take the next step towards in vivo. If we can help people understand how to navigate the use of cell-free systems in their various manifestations from reconstituted to lysate-based as an enabling part of a design, build, test, learn cycle, I think that would be, I think that would do a lot for folks who are trying to accelerate their development.
There's another one here around, you sort of mentioned at the start of your talk around these kind of control systems and building kind of inherent robustness by having kind of feedback within the system itself. I was just interested to know what kind of feedback control mechanisms are you looking to implement? And you mentioned also about computation. Are there things that you're doing in those control algorithms that are maybe different because it can be done in the biology as to what you might do with, say, an electronic system that you're building, and whether you're taking any inspiration from natural feedback control type systems that already exist?
That's a great question, and it's a whole long discussion in itself, of course, to be had there. And so currently we're working towards implementing control. Right now we're focused very much on sensors and cellular computation, which we hope to later take into some of these control strategies. When I was at DARPA, I led the biological control program, as you know. And one of the most fascinating parts for me about putting that program together was getting people in the room who were experts in control theory in a very rigorous way, and then bringing them face to face with the biologists and saying, okay, what can we do? What can translate? What doesn't map on? What needs to change? Because I'm hoping that, like, and you see this too in what people are doing with cellular computation, where everybody's like doing Boolean logic. Well, no, like we only do that because that's what you need to do to get published. Quite honestly, like nobody thinks it's a good idea that I can tell. And so we're very eager to rapidly move to other ways of doing cellular computation. For example, Lynn, you have different classifiers that you can implement. And so we're kind of looking to machine learning in silico for some inspiration there. But I think ultimately, hopefully what we can do is understand biology enough on its own terms that we can bring more biological ways of computing and back over to in silico computing. There's got to be a feedback. I think there's a lot of potential there for really interesting advances. I know that didn't answer your question at all.
No, that's what gets me excited as well. Is this? It's that intersection and figuring out what translates and what doesn't, and that often tells you a lot about the constraints on the biology as well, which I think is super important to understand and to work around and all of that sort of thing.
And one thing I found very exciting personally as somebody sitting at a metrological institute is that oftentimes, as your measurements get better, you're able to see the ways in which your assumptions are breaking down or your system itself isn't really functioning ideally or the way you had intended. And that's exactly where I think you need to drill down and understand, because that's the system, that's nature trying to tell you something.
Definitely. Right, I realize we've reached the hour. So Lucia, do you want to ask the last question, then I'll wrap things up?
Yeah, very quick one. So there is a question about how do you think that bottom-up approaches would compare to build minimal cells as compared to the top-down that the GCDI uses?
I see them as they're of course very different, and today they're very different, and they're both absolutely necessary and entirely complementary. Because when we draw that spectrum from chemistry to biology, there's still a point in the middle somewhere where all we can say is that magic happens. And I hate that word magic, personally, as a scientist. I really dislike it deeply. It really, here there be dragons. And so I'm hoping that, and that's one thing I like about the Build-A-Cell community, is it does bring together the bottom-up folks and the top-down folks into a sustained conversation, so that hopefully together we can start mapping out that unknown territory and understanding this gray zone, because I don't think it's a single point. I think it's a spectrum from definitely not alive to definitely alive, and in between it's, we see this with viruses too. Are they alive or not? So I think we'll start recognizing a lot of naturally evolved biology in that space as well.
Thanks.
Awesome. Well, I want to say thank you again, Elizabeth, for a really wonderful talk and for sort of opening our eyes, I think, to a lot of the things that are going on at NIST that I suspect a lot of people just weren't aware of. And I also want to thank Lucia also for doing a brilliant job with fielding the questions. And I hope to see some of you back here again next month. So thank you, everyone.